Normal University, investigador postdoctoral en el Instituto Max Planck de Colonia, Alemania. Es director e investigador principal de la Universidad de Burdeos, Francia. Investigador invitado en el Boyce Thompson Institute de Estados Unidos, miembro del equipo GENEPI, Diversidad Genética y Epigenética de las plantas de uva y su contribución relativa a las variaciones fenotípicas y adaptaciones al medio. Ha dirigido numerosos tesistas de maestrías y doctorados, como también integrado el Comité de Evaluación en Biología Integrativa. Ha sido miembro del Consejo Nacional Francés de Universidades y asesor en distintas instituciones de nivel superior. Registra numerosas publicaciones como autor y en coautoría, tanto nacionales como internacionales, en temas de su especialidad. Es co-coordinador por la Universidad de Burdeos y responsable de la implementación de 3 módulos en el marco del proyecto Vita Global. El doctor Galucci tiene una extensísima ruta de vida académica que hemos sacrificado en esta presentación en honor a la brevedad del tiempo. Bueno, Philippe va a hablar puntualmente de epigenética. Primero va a hacer una breve introducción sobre qué es la epigenética, va a explicar el tema y después va a explicar cómo se aplica a la vida real. La presentación va a ser completamente en inglés, cada determinadas diapositivas vamos a ir parando, así bueno, cualquier duda que vaya surgiendo, vamos haciendo un pequeño resumen y continuamos con la clase.
Je peux le faire en français aussi. We can vote. So, first, thank you for inviting me. Thank you, Rimena, for organizing all that, and Nicola and Natalia as well. I'm going to speak of different things. The first will be a rather long introduction on what is epigenetic and what are the different concepts around epigenetics. And then I will take 2 case studies in grape to explain how in my group we have been studying this epigenetic regulation, how this has helped us to solve, some difficulties we could have facing, grape physiology. So what you see on this slide here is ISVV. This is the place where we're working. It's a rather big laboratory. We are 500 different researchers. And we mainly focus on grape wine in a vertical aspect, which means we start from the plant and go to the field, including commercial aspect, everything. So everything is covered in the field of viticulture. So the lab I'm working in is called EGFV, which means functional genomic and ecophysiology of grape wine. So we study both the gene function, but also what happened to the field when we transfer the plant into the field with different types of experiments. So this will be the general organization of the talk. You tell me if I'm speaking too fast. I'm trying to slow down, but I have a tendency to accelerate when I'm speaking. So please tell me. So first will be what is epigenetics. So this is going to be quite long because I don't know from what I have to start. And then I will explain how this epigenetic is one of the major actors of what we call plant memories. And then I will have, as said, 2 case studies, one related to a trunk disease, which is named SK, and which is a problem in France, actually. And then we had another study where we were focused on carbohydrate limitation into fruits, into grape wine, and see how this impact on epigenetic. And finally, I will have some concluding remarks. So let's start by what is epigenetic. So here you see a couple of examples. Some real. This is a real picture of clones. Cow clones that should be strictly genetically identical with the same genome. They will multiply. And then you see that still they do present differences. Not major differences. Obviously, they are just the color of the skin, but still they do have it. Here it's a funny picture of 2 twins. And they're quite old. You see with age differences have appeared. And then probably the most famous picture of Dupont, and you see they are twins too, but they are different. If you look at the mustache, then you see there's a slight difference in the mustache here. So So how can this be explained if everything is strictly identical at genetic level? So that's where, I mean, people are starting to speak of epigenetics. And this was to show that this has also gone into the normal societies, not only scientific story, but there has been a lot of articles where people have started somehow 10 years ago to realize that there were more than genes to define an organism, but the way these genes were used. There was some even funny articles telling now you can take power over your genes, which mean by your habit, the way you behave, whether you smoke or not, whether you eat fat or not, whether you do sport or not, this will impact on the way that your genes will be used, and not at the moment of you do it, but there will be some memory of what you do over time. Sorry. Oh, there's something strange here. Okay, so that's the correct one. So let's start with the very basic. You all know that. So in each cell that makes an organism, you have DNA. DNA is all the information you need to have the cells functioning, the organism build it there. But the DNA is highly organized, and you all know that you have interactions between specific proteins, named histones, to create a highly complex structure called chromatin. And this chromatin itself will be organized in a highly complex way. You have different level of compaction. This is the maximum level. What we call here, it's a metaphasic chromosome. This only occurs at the cell division, but whatever. So this is very dynamic from this state, which is almost totally open, like a bead on a ring. And this state, you have many mechanisms that control and compact the chromatin. So all this is in one cell. So imagine you have your DNA is 2 meters long. I mean, one molecule of your DNA is 2 meters long, and all this is in a few micrometers within your nucleus. And all cells are like that. So that's the basis of each organism. And this is also where what we call genetic information is stored. Yeah. Can you give me a second, please?
Bueno, ¿cómo vienen hasta acá? ¿Alguna duda, alguna pregunta? Hasta ahora lo que está haciendo Felipe está dando una pequeña introducción antes de entrar a la definición de epigenética. Bueno, lo que hizo primero planteó que hay individuos que son iguales genéticamente, pero también tienen diferencias, dio el ejemplo de los mellizos, y ahora está dando una pequeña explicación sobre genética básica, con la estructura del ADN, los cromosomas, y cómo se componen esa parte de la genética.
Is it okay?
Perfect.
So we can try to see this in a different way. Maybe it's you. So just to point out, to see it in a different way. So this is still the same chromosome, and here you have the DNA, and of course on the DNA you have a lot of different information. If we just focus on the one we usually take care of, which are genes, you can see this as books. So you have all over your genomes, books scattered, sometimes all together, sometimes separated. And then you can represent this like, so small libraries. One chromosome is one with thousands of books, and the other one, and so on and so on. All our genomes is like that. So you have, and theoretically, all of the genes can be read. You can have access to all of them. In theory, at least in what we call, stem cells, this is what happens. So theoretically, any gene could be used. Practically, during differentiation of an organism, you will have cell type, specific cell type, which appears. And as you see, each of one, this is in plant. Human like this would be strand. This is more in humans. And if you look at it, they have very specific shapes. And these shapes can be explained by the fact that it gets specialized. And there are many reasons why they get specialized, but one of those reasons is that some of the gene will be functioning, some other will not be. And this is defined very precisely. And so the question is why some of the genes, I mean, they all have the same genome, the same chromosome, but if you look at this, in some of the cell, this will be functioning, the other will not be. I can show it in a different way. So you have one cell phenotype here, another cell phenotype here. This is the chromosome, the same chromosome 1. This cell is able to use this part of the chromosome, not the other parts. And this cell is able to use all this part of the chromosome, not the others. In other words, they have lost the keys to open the other part of the, okay? So which means, how is it possible that they lost the keys? And this is very rapid. It's just one decision when the cell differentiates at one stage. It's fully potential. And then one decision later, part of the chromosome will be closed.
Bueno, esto que está diciendo Philippe es importante. Lo que plantea él es que las estructuras de los cromosomas se ordenan como libros, que todos estos genes se pueden leer. Las formas que estuvo mostrando Philippe de las estructuras celulares dependen de genes específicos, y que si se pierde por ahí alguna parte del cromosoma, se manifiesta como una expresión de la estructura vegetal diferente. ¿Es así o no? Estoy medio...
Va a tomar esa célula, toma y la planta, lo que le pasa es que de toda esa biblioteca que tenía los mismos genes, toma como preponderancia una parte, y entonces eso es lo que se expresa, eso es lo que se diferencia en este caso en una célula del estómago, y pierde la capacidad de usar el resto, o de que se exprese el resto. Entonces, lo que está diciendo es que el cromosoma es el mismo, y lo que está puesto en más claro es lo que se está realmente usando en un lado y lo que se está usando en el otro lado, y por eso el fenotipo termina siendo diferente. Y entonces dice, bueno, ¿cuál es la clave o por qué esto sucede? Porque una vez que esa célula se diferenció en algo, me es más fácil en un tejido humano, una vez que se diferenció para formar parte del estómago, no, esa célula no forma parte del corazón. Entonces, ¿cuáles son las claves para que eso suceda?
Of the chromosome are locked and cannot be used, and another says other parts are locked and cannot be used. Okay, so there's a physical reality. I mean, this is a schematic representation of what is really in a cell. So the part which is not accessible usually is called heterochromatin, which is highly compact chromatin. And the part which is accessible like this, light gray instead of black here, is called euchromatin, for true chromatin. So we can also represent it like this. This is open, so euchromatin, and this is closed, heterochromatin. I will use this representation from now. So, of course, you have a lot of level of condensation in the chromosome. So you have the basic one, I mean, the one you see maybe sometime on microscopy, which is called beads on a string. Then you have a first level of condensation, then a higher level of condensation. Then this is heterochromatin, and this is the metaphasic chromosome. And all of this, I mean, if you look at the gene, whether you can read them or not, here you can, and here you can't. So I put it in a different way, the same thing but said differently. And so if we summarize all that, so what is the genome? The genome is the information we all have. This is the stored information. This is what makes us. But then this information is organized. So the stored information is organized in chromatin and higher level of organization, which can be very high. And these levels of organization determines what part of the genome can be used. So if it's closed, you don't have the keys, then it's in red here, you don't have transcription, you don't have gene expression. If it's open, like in the traffic lights, then you can have transcription, the gene will be functioning. And this will define, I mean, the combination of genes that are expressed and not expressed will define the cell phenotype and eventually the organism phenotype. Of course, this is not spontaneous. You have biochemistry behind that. And the biochemistry behind that, I just focus on the two main aspects, but there are many others, but the two main are the fact that when you look at DNA, so DNA, I remind it's a succession of, let's say, bases, adenine, cytosine, guanine, and thymine, the cytosine can be methylated. So you will find in the genome, any genome of eukaryote, cytosines or 5-methyl cytosines, which are mixed all together. And in addition to that, you have also a lot of information just on the other part of the chromatin. You know, these proteins that I call histones, they have a kind of extruding part here, which is a bit of helicoidal, and this can be post-translationally modified. So you have here phosphorylation that can occur, acetylation, methylation, ubiquitination, and all this also is considered as a signal that would control the level of condensation of the chromatin. Maybe you can.
Lo que se denomina genoma es justamente la información que está contenida en el DNA, en el ADN. Pero luego nosotros tenemos a su vez la cromatina, que es la parte que está, digamos, como condensada ahí y que el resultado de... Tenemos en esas flechitas vemos lo que dice represión y lo que dice transcripción. Lo que dice represión es toda esa parte que estaba como oculta, que estaba como que no teníamos la llave en la filmina anterior y que no se expresaba. Y lo que sí pasa por la transcripción es efectivamente los genes que pueden expresarse y la relación que hay entre lo que está reprimido y lo que se puede transcribir me va a mí a dar el fenotipo, básicamente, que va a tener esa célula. Pero ¿por qué pasa esto? Digamos, en el tema de la represión, ¿qué es lo que puede pasar para que de repente se transcriba o no se transcriba? Dos procesos principales. Uno es la metilación del ADN y otro es la modificación de las histonas. En cuanto a la metilación del ADN, a la derecha está explicado cómo se produce y es a través de una de las bases del ADN que es las citosininas. Las citosininas tienen la capacidad de metilarse o de no metilarse. Entiendo que de acuerdo a cómo se metilen y a cuánto se metilen, bueno, van a hacer que eso pueda o no transcribirse. Y después hay otro nivel en el ADN que puede ser modificado y que son las histonas, que son esas cositas redonditas que se ven ahí. Y esas tienen distintos procesos de modificarse. Los nombró, pero no los recuerdo muy bien. Una era fosforilación, acetilación, metilación. Bueno, tienen distintas posibilidades, siempre, ¿no?, uniéndose a otro compuesto, de modificarse. Entonces, esa parte que no teníamos la llave para poder abrir, digamos, mediante esos procesos, sí puede producir cambios, y entonces la combinación de todos esos procesos me hace, y por eso abajo, que en definitiva yo pueda terminar teniendo con un mismo ADN, porque el genoma es el mismo, distintas expresiones de los genes, distintas serían resultados fenotípicos. ¿Está bien? Sí, perdón. ¿Quiere decir algo?
I have a question. Sorry for my ignorance. I belong to the management sciences, but as you said, this is information that is lacked. Is there any possibility in the future to access, to get available this information, maybe for any kind of development or evolution?
So, there's a very good question. So, when it's locked, can this be unlocked? Yes. Not always, but yes, depending on various external parameters. And sometimes when it's unlocked for bad reasons, this can lead to misfunctioning of the gene or functioning of the gene at the wrong time and lead to illness. And it's basically because of cancer in some situation. So this can be logged, or it can be logged when it should not be. But it's more simply than the development in this case is interaction between the development and environment. Okay, so this is what we consider a gene, but of course this occurs on the whole genome. And we have methodologies. I will maybe explain briefly one later. Also, we have a methodology that allows to look at the whole genome, the distribution of this methylcytosine, for example, where they are, and we can position them at all genes, all positioned on the genome, and the same for this post-translational modification of histones. So we can check for each cell type of tissues at least whether, we call that the epigenetic landscape of a cell, so which mean or the epigenome, so which means exactly the distribution over the entire genome of these different epigenetic marks. And this is one aspect I will show later how we used it. So to summarize, we have two different possibilities of situation: open genes. So you have the keys, closed genes that don't have the keys. So you can read, you cannot read. And reading a book means reading the gene, so producing mRNA. And if you produce mRNA, you eventually produce proteins behind, and then you create a phenotype. So it is basically 20 minutes to say all that. I could have started only by that. No? Okay, you don't need to translate probably here. But yeah, last point, and this finally answer your question. This, balance here between these two situations is of course developmentally controlled if life was perfect for all of us. But of course you have a strong interaction with the environment that can interfere and redefine in some, and eventually this can be inherited.
Es la letra en verde, la que dice on, que bueno, tiene que ver muchísimo con el desarrollo, pero también hay una influencia muy grande del ambiente y que en definitiva termina en que esa parte del ADN es la que se puede leer. Hay producción de ARN mensajero, y por lo tanto ese ARN mensajero es el responsable de la síntesis de proteínas en definitiva, que es lo que termina produciendo un efecto. Y hay una parte que es la que está locked, la que está cerrada o bloqueada, que es la que no me permite, yo no la puedo leer.
Okay. So then we can ask, is that real? That is, that's really the impact on phenotypes. So I've taken here a couple of examples that we published some years ago that show that this change, not in the DNA sequence, but simply on the methylation statue of, some genes on some cytosine, can lead to highly stable and transmissible phenotype. So probably the first identified here on Snapdragon is this change in the symmetry of the flower. You have here bilateral symmetry of the flower, and here you have the central symmetry of the flower. And so this is a very strong phenotype, and the gene has been isolated, identified, and when you compare the sequence of the genes, they are strictly identical. There's no gene difference. They're the same genes, no differences. But when you look at the methylation, which is the methylated cytosine I just spoke about before, then you find out that on one of the genes, some areas are hypermethylated. So you have an increase in methylation, and this locks the gene. And by locking the gene, you change the phenotype. And this is behaving like a normal mutation. So you have a classical, what we call classical Mendelian segregation through generations. The same for this one. This is a dwarf mutant in rice. And these are the one which is on tomato affecting the color. It's not the color, it's the ripening of the fruit, which is called the CNR, which means colorless non-ripening. So this is again the same story. It's a very stable phenotype that has been transmitted over tens of generations. slightly reversible. I'll come back to this point later. And when you clone the gene, the same. I mean, the genes are strictly identical. There's no mutation at all, except for this cytosine that are methylated into one part of the gene. And this causes silencing of the gene and the phenotype.
Muestran lo que acaba de explicar antes. En todos los casos estamos hablando de individuos que tienen exactamente el mismo genotipo, pero no hay diferencias, ¿no es cierto?, en el genotipo, pero hay diferencias muy claras en el fenotipo. En un caso el tema de la flor, en el otro caso uno que es enano y el otro que es normal, y en el caso del tomate uno que como que ha perdido la capacidad de madurar y tomar color, y el tomate normal. Y lo que se ve cuando se hace el análisis es que si bien el genotipo es igual, lo que no es igual y lo que ha cambiado es la metilación de las bases, y eso hace que el determinado gen, que es justamente el que está metilado, se bloquee. Hay una cruz ahí, se produce el bloqueo de ese gen. Y eso es lo que produce el cambio en el genotipo. Y lo que es interesante es que ese carácter, si bien no es una modificación genética, se transmite con las generaciones. Entiendo que en el caso del tomate dijiste que hasta 10 generaciones se transmitía ese patrón.
It's a classical Mendelian segregation.
Que es una clásica segregación mendeliana.
So that's why it
was cloned by classical chromosome cloning. So yeah. Just to show you, this is some work we have been doing in the lab, and we have just modified the ability of the plant to remove methylation. So you have mechanism. I don't explain it because it would have been too long, but all plants and all organisms have mechanism to put methylation on and then to remove this methylation. So here in this experiment, we have impaired the ability of tomato plants to remove methylation. And the idea was to check whether this had an impact on ripening. So these here are two different transgenic plants that cannot demethylate efficiently. And first thing you see, they cannot ripen efficiently. And furthermore, they have also many developmental defects. So the fact that you interfere with the ability of an organism to control precisely during development its methylation level at specific loci or everywhere will cause very dramatic phenotypes. And sometimes, and this is another plant in Arabidopsis, if you impair the ability to set up methylation, then you kill the plant after 1 or 2 generations. Actually, in tomato, you cannot impair to set up methylation, you kill the plant immediately. So these are other phenotypes of this variant. You see we impact plant size, leaf shape, flower shape and organization, leaf organization, and fruit, flower and fruit organization and shape. So everything is affected. So this shows that it's a highly important mechanism, at least in this plant for this process.
Haciendo lo que muestra esas imágenes, que en realidad la planta también puede demetilar lo que tenía metilado o lo que se había metilado. Entonces, ahí está mostrando, bueno, distintas variantes, distintos resultados de distintos grados de metilación o no, de eso que se había metilado. Y ahí están todos los distintos fenotipos que pueden darse de acuerdo a cómo lo ha la cantidad o que se ha demetilado o no, y que eso tiene consecuencias, se ve que tiene consecuencias muy, muy importantes en el resultado, porque en el caso de los tomates o en la Arabidopsis, en 1 o 2 generaciones se podía llegar a matar al individuo, si eso seguía ocurriendo.
Okay, so far we have not been speaking of memory, we're just speaking of basic mechanism of chromosome organization.
Bit of biochemistry behind and how this could impact plant phenotypes. So the question now I want to discuss is there a link, and what link between epigenetic and memory, cell memory? I'm not speaking of brain, I'm speaking of cell, I mean basic and fundamental memory. So this part. So first thing, plants have memories. Don't do stupid things to your plant, they will you will be rewarded. And so this has led people to think that finally plants are intelligent. They have an intelligent behavior, which means they are able to learn, to store the information, and to reuse it. Not in the same way we would do it, but still they do that. And this is illustrated by the fact that one can acclimate. Acclimate means you have been subjected to something, and then you will react differently to the same thing later. You better adapt it. So which means plant may memorize part, not all, they're like us, they forget a lot, luckily, and then they can remember part of the past. And also, in some cases, they can transmit this to the offspring. So this is highly controlled, but part of it can be transmitted. So the important information is that the memory, which is the ability to reuse the stored information in plants, relies mainly on cellular, molecular, and biochemical networks. There's no brain, there's no way of thinking, but this way of learning, the cell learns through different biochemical mechanisms, including the epigenetic process. But it's not only epigenetic which mediate memory in plants. Oh, sorry.
Bueno, lo que está explicando es cómo relacionamos la epigenética con la memoria de las plantas. Entonces, empieza introduciendo que bueno, las plantas pueden aclimatarse, que significa que se adaptan a un ambiente que está en constante cambio para poder optimizar su fin, para poder estar, digamos, seguir estando bien o estar en forma en ese ambiente que va cambiando permanentemente. Las plantas pueden memorizar parte, no todo, de lo que les ha sucedido en el pasado. Cuando hablamos de lo que les ha sucedido es básicamente la acción que el medio ambiente, su entorno, que ha tenido sobre esas plantas, y que lo que es interesante es que eso lo pueden transmitir, es decir, lo que han aprendido, esa adaptación, que han aprendido de experiencias anteriores, lo pueden transmitir a las generaciones siguientes, es decir, a su descendencia. No todo, parte de lo que han aprendido. No es, y que me imagino que no sabemos qué es lo que van a transmitir y que no lo podemos predecir. Y que en realidad en las plantas, que claramente no tienen memoria como nosotros, este aprendizaje en realidad se basa en la habilidad para reusar la información que tenían almacenada y que está basada exclusivamente en redes celulares, moleculares y bioquímicas. A través de esos procesos es que se genera esta memoria. Felipe, can you speak with the microphone?
So just take one example of memory, which is the priming in plant, priming system. Now it's too loud, no, isn't it? So what we call priming in plant, the fact that a plant that will be submitted to a first stress, then recover, will react differently to the same stress later. So this is typically the idea that the plant has memorized the first treatment, and then, thanks to this memory, will be able to not perfectly live, but to live in a much better way the same stress. So this is called priming, and is more and more used in many of the application in agronomy. So what it is due to? There are different explanations. One aspect, and I will come to some others later, one aspect that when you prime your plant, you may trigger one response, which is either faster. So when the plant has been primed as compared to the non-primed, the naive plant, we say, if you look at the response to stress, can be in the prime plant either faster or stronger, or could react earlier, so which is more sensitive to the stress. So whatever the mechanism is, the answer is that the plant will behave in a better way. So you have different types of priming, of course.
¿Cómo se produce esta memoria? Bueno, ante una exposición, la primera exposición a un estrés, en este caso una deshidratación, y luego algunas plantas se pueden morir, otras no. Bueno, ante una rehidratación, esa planta que ha sobrevivido a una segunda exposición a la deshidratación ya no se va a comportar de la misma manera. Es decir, que tiene una memoria, ha podido construir una memoria de ese estrés y va a comportarse, como muestra el dibujito, de una manera como un poco más fuerte, está como mejor preparada, ¿no es cierto?, para enfrentarse a ese estrés. Entonces, ¿esto cómo funciona? Tenemos dos tipos de planta, la que ellos denominan la prime, que es la que ha sido sometida por primera vez al estrés, y la naive, que es la que no ha sido sometida. Y esa exposición al factor adverso, al estrés, etcétera, puede desencadenar distintos tipos de reacciones. Una reacción que puede ser muy rápida y temprana, que es ese tipo de gráfico que se muestra arriba. Después una muy fuerte, en donde claramente la curva cambia y al principio es como menos pronunciada y después tiene un pico de reacción, y una que dice y una sensibilizada que tendría esa forma. And what is that you have above alter?
I didn't present on that.
Okay.
This is the network that can be achieved in time.
Okay.
Okay, so
Another way to say the same thing. So when a plant will be attacked in some way, will react. So the reaction, so far I've been speaking mainly of that, but indeed you have many other things occurring. You have change in metabolite level, you have change in protein, post-translational modification, fine hormones have involved, transcription, transcript and mRNA are involved. All these, can participate to the memory. What changes between the different processes is a time scale. Some are very short-term memory, like metabolites or some accumulation of transcript. And some others are rather long-term memory, which is what I will focus on, which is, epigenetic is more long-term memory, while the other, although this can be discussed, but make it simple, this is rather short-term memories.
Cuando ocurre algún factor estresante, bueno, ahí en este caso he puesto granizo, sol, abióticos, un insecto, hay distintos mecanismos adentro del organismo que se desencadenan, es decir, como que la reacción puede estar desencadenada a distintos niveles, desde, por ejemplo, acumulación de factores de transcripción, cambios en los niveles de hormonas vegetales, modificaciones post-translacionales y cambios a nivel de metabolitos. Pero, de acuerdo a cuál de esos se exprese más o menor, es la velocidad del cambio y también el nivel o la duración que va a tener esa memoria. Sin lugar a dudas, los cambios que se producen en la metilación del ADN y en el patrón de la cromatina, es decir, los dos que habíamos visto antes, estimo que es lo que va a explicar ahora, son los que perduran. Yo ahí puedo poner un ejemplo rápido. Cuando una planta de vid, por ejemplo, nos sometemos a estrés hídrico, es decir, que le empieza a faltar agua en el suelo, rápidamente lo que hace es un cambio a nivel de hormonas de crecimiento. Rápidamente las raíces detectan la falta de agua en el suelo y sintetizan ácido abscísico, que es una hormona natural de las plantas. Es decir, so it's another thing, I am explaining another thing. Y eso hace que se cierren los estómagos, un cambio como inmediato, pero eso no se transmite.
So another thing which is quite important to understand here, so memory is costly for the plant.
So if the stress is not coming soon again, so the plant will forget, and it's safe for the plant to forget. So you always have this balance between the memory and forgetting what the plant has been facing. And this can be, this trade-off is very important for maintaining, the fitness of the plant. So now let's speak a bit more in details. I think this is memorized. In fact, when we look at DNA methylation and chromatin changes, you have two situations, so which are summarized here. In one case, in the same situation, you have an elicitor, the chromatin state changes, and then you remove the elicitation, come back, and everything is reversed. So this is not really the topic of epigenetics. This is just a classical chromatin regulation. Here you have no memory. In the same situation here, same signal, you will go to a change in chromatin, the same, but then the cell divide and you maintain the imprint, the epigenetic imprints. And in this case, you have a memory. So this is really what we study in epigenetic. We don't study that. This is classical gene regulation, chromatin changing, you have an impact and then you look. We study the fact that when the signal has been removed, then the cell maintains the modification it has, and this is real memory. Yeah.
Cromatinas pueden tener dos tipos de vías. Cuando se produce el efecto adverso del medio ambiente, la señal puede no transmitirse o sí o transmitirse. En el primer caso no se transmite, es decir, como que se produce el cambio, pero luego cuando esa célula se divide no se transmite. Entonces vemos que tenemos la misma célula que teníamos antes del estrés. Esa no hace a la memoria de las plantas. En cambio, en el caso de abajo no pasa lo mismo. Pasa que el cambio que se produce sí se transmite, y entonces ahí es cuando hablamos de epigenética y ahí es donde se estaría produciendo la memoria.
Okay, so what is very important to understand here, epigenetic is the maintenance through cell division of what has been modified at any step. So I will put it in different way here. But even when we speak of memory, we have to consider many different situations. The simplest is you take one organism, one plant, from baby plant and then growing. And so let's say it is in paradise, this plant, everything is perfect. So the plant will develop, organize its chromatin, and then you have cell divisions. This is what we call somatic memory of the cell. During cell division, the epigenetic landscape that has been defined are maintained. And at the end, you have a plant which is normal. And then you apply a stress, and the plant will react in a specific way to this stress. So this is the first situation. But you may imagine that the plant is suddenly transferred from the greenhouse, the paradise greenhouse, to the normal field. Then it's hell for it. Then you will have a lot of triggering, and this will prime the plant, as I explained before. Then when you put the plant in this situation, it will maintain part of the priming situation, and then you still have a somatic inheritance on this that will lead, when you have a second stress, to a reaction which is different from the one of the naive plants. So all this is what we call somatic memory and is mediated by the activity of the meristems.
Bueno, a ver, este es un poco más difícil. Esto está explicando qué es lo que sería la memoria somática. En el primer caso tendríamos una planta en el paraíso, supongamos que la tenemos en un invernadero. Entonces esa planta, cuando se produce una planta normal y cuando se divide, en el primer caso tenemos esas situaciones, plantas que son iguales y después otras que no son iguales. Pero si a esa planta que la teníamos en el paraíso de repente la ponemos en el campo, en donde tiene el sol, el frío, el viento, es decir, como que hacemos el prime, que él había dicho, que es la sometemos por primera vez a un estrés, luego va a producir una planta que cuando segregue no va a ser igual que la que tenemos del lado izquierdo. A eso es lo que denominamos memoria somática. Entiendo que ha podido transmitir, a la otra generación el estrés, la memoria que había adquirido.
It's only one plant. It's only one plant through meristem. No transmission.
Perdón, es una sola planta, no hay segregación acá, es una planta que se produce agámicamente, digamos.
There's no sexual.
No hay división sexual, son todas reproducciones agámicas. Por eso se llama somática, claro, sí. Por eso es memoria somática.
Okay, so that's one thing. Then the situation becomes more complex when we go to the next generation. And there's two ways to do that. You have sexual reproduction, classical. You have two gametes formed. So first, when you have gametogenesis, then you lose some information, you reorganize it. So what is in the gamete is not exactly what was in the parents. And after sexual reproduction, we know that mainly through the female gamete, we have some epigenetic transmission of what was in the parental lines.
Bueno, después en cuanto tenemos dos formas de reproducción, bueno, la sexual, en donde se pierde parte, ¿no es cierto?, de la información, pero se sabe que gran parte de lo que se obtiene proviene de los gametos femeninos.
Sorry.
Sí, dijo eso, que proviene en realidad de la parte de female, proviene, ¿no es cierto?, de los gametos femeninos.
Then the second type of, reproducing plant is non-sexual. We call it agamous propagation. It's typically what is done in grapevine, for example. And then it's very different. You don't have gametes formation, you don't have meiosis, you only have meristem propagation, so through, mitosis. And of course, you have things occurring because you have a rejuvenilization of the plant, which might erase part of the information that was provided in the older plant. But still, most of the information should be maintained because you just have mitosis, and so it's cell division, classical cell division. So in this case, and there are mainly work, in trifolium repens, so clover, that show that part of the information is strongly maintained when you do this type of reproduction.
Bueno, y la reproducción agámica, en donde claramente no hay meiosis, no hay gametas de por medio, sino que se reproducen los meristemas, teóricamente debería pasarse toda la información que tenía la planta madre, digamos, pero hay algo que se pierde porque hay un proceso de rejuvenilización, entonces hay algo que se pierde, pero teóricamente no estaríamos en la situación de estos signos de preguntas que cuando hay un intercambio de gametas uno no sabe cuál va a ser el resultado, sino que deberían mantenerse las características de la planta madre. Que es el caso que nosotros usamos, claro, para propagar la vid.
So this is what apply to grapevine in general, more complex because then you are grafting in addition, and so you have this process for the scion and for the rootstock at the same way. But then grape is also a perennial plant, so which means you will grow the plant 1 year, it will be subjected to different types of stresses or not, and then winter comes. After winter, you might have the question, what happened here? You have a complex situation because you have preformed leaves the year before that were subjected directly to the stress that when they were formed, to the stress of the year before. But then you will have neoformed leaves that will appear later, and which are the result of the meristem functioning. So these leaves, not the first they have been grown here, but these leaves may have a type of memory mediated by the meristem when they grow, so much later. And when you compare, and this is part of the thing I try to explain later when you compare these neoformed leaves, the question will be, do they have the same epigenetic landscape than the same leaves the year before? In other words, what part of the year before treatment that has impacted the meristem will have consequences of the epigenetic landscape of the neoform leaves? Is there some memory there?
Bueno, acá lo que dice es que bueno, la vid es un cultivo perenne, es una planta perenne, entonces que está sujeta, ¿no es cierto?, a la acción del medio ambiente y de los factores externos. Tomemos en una temporada. Nosotros sabemos que al año siguiente la vid tiene una parte que estaba cuando brota, ¿no es cierto?, esa yema, una parte de las hojas, un número de hojas estaban preformadas del año anterior, normalmente son entre unas 6 y unas 8, pero después hay toda una cantidad de crecimiento que es el neoformado, es el resto de los nudos y de las hojas que se forman a partir del meristema que no estaban formados del año anterior, sino que se forman en esa temporada. Entonces la pregunta es, ¿esas nuevas hojas que se forman tienen la memoria de lo que había ocurrido durante el ciclo pasado? ¿Sí?
Sorry, I realized it's not the last. This one shouldn't be there. So I would try to illustrate this through one first case study. So we have been interested in studying, the SCAR illness in grape wine, which is a major threat in our region actually. So you might know this is a trunk disease that affects the vascular bundles, and here, for example, they lose color. There's some hydraulic failure into the plant, and this can lead, of course, to the dying of the plant. And it has a very precise. This was a collaboration with Chloé Delmas here, she's a specialist in that, and it has a very precise phenotype that the leaves, and I come here to this one, the leaves will present after when they're grown. At the beginning in my country, in the beginning of July, they will start, to dry all over, while only the internal part will remain green. But the interesting thing for this illness is that all the plant is not affected in some way. Here you have a typical case where half the plant will develop a symptom, and here you have another sector of the plant which appear totally normal without any effect as compared to the control plant, which in this case was located 5 meters away, so it's the same lot. All plants that we have been harvesting, are exactly in the same place, and some will be like this, very, very nice, and some will be like this, some will die after. And so the idea we had here is, can we analyze the consequences at all levels? The strategy. I'm sorry, this is not the final, I don't know why, it's not the final presentation I've been preparing. I don't know where it is. Okay.
Do you want to?
No, it's fine. I will manage it. So the idea is to accumulate different levels of analysis on this type of plants. So the first level will be simply the phenotypes. So we want to control over years, and these plants have been monitored during several years. We want to control the phenotypes when they appear, where they don't appear. The second thing we want to do is want to look at the metabolites. The metabolites are the ultimate point when you look at a plant. Metabolites are the result of all that has happened before. And then third thing we want to integrate are the transcript, because transcript is what determines what proteins will do, and the change in transcription is very rapid most of the time and gives us an overview. And the final point, I just finished my sentence, I saw you. And the final point is we want to relate all this to the epigenetic regulations and memory.
Lo que va a explicar ahora es un caso de estudio que han estado estudiando en Francia relacionado a la enfermedad de ESCA, que es importante en la región de Burdeos. Entonces, lo que está mostrando, es que es la enfermedad que tiene esa sintomatología típica de que se pone necrótico y de ese color rojo amarronado el borde de la hoja y el centro queda verde. Se ve que no lo presenta toda la planta, es decir, hay una parte de la planta en donde presenta el fenotipo, la sintomatología, y la otra parte no la presenta. Entonces, ellos empezaron estudiando esto en distintos niveles. El primer nivel era, estudiar el fenotipo, es decir, cuándo está y cuándo se presenta y cuándo no se presenta. El segundo nivel es estudiarlo a nivel metabólico, es decir, cuáles son los metabolitos que se producen en las dos condiciones, ¿no es cierto? Claro, en las dos condiciones. El tercer nivel de estudio es el nivel transcriptómico, ¿sí, Filip? A nivel transcriptómico. Y el cuarto, perdón, ¿puedes repetir?
Epigenetic.
Y el cuarto ya es a nivel, claro, a nivel epigenético, es decir, que ese nivel transcriptómico, digamos, cómo está relacionado con la epigenética de esos individuos.
So for this first experiment, I'll go to the next. So for this first experiment, we used leaves that had already developed symptoms. So we are quite late. We are not using newly formed leaves. We know that these leaves have symptoms. These leaves on the same plant do not have symptoms. So SK symptomatic, SK asymptomatic. No. And then we have a control plants. I mean, the neighbor, which is lucky, no symptoms. And the idea was to compare on the different parameters that I've explained before, to compare the three situations. So the same plant, those having symptoms to those that have no symptoms. Two different plants, no symptoms, and two different plants, no symptoms, no symptoms. The idea is to find out what are the differences. Can we discriminate easily this from this? This, obviously, we expect that this and this will be very, very different, but from that, we don't know at all, because they are different plants. One is sick, the other one is not, even if there's no symptoms here. So these are the different things we have been integrating, and this was a project managed by my previous PhD student, Margot Berger, and a postdoc in my lab, and Bernadette Rubio. So what are the differences between, and the basic question we had, we're in the same field, plants are close to each other. Why do plants develop symptoms and some other not? What are the differences between these plants? Can we find that?
Bueno, eso lo entendieron, era fácil.
It's okay?
Sí, sí, okay.
So I don't go through all details. Simply, this is a principal component analysis of either the metabolite profiles or on the transcriptome of the all gene expressed into a cell. So if you look at the metabolites here, so this represent the symptomatic leaves. You see they all group all together, and all the non-symptomatics, whatever the plant of origin, group all together. In other words, we cannot distinguish leaves on their origin. They are exactly the same at the metabolic level. So when a leaf is functioning, the leaf is functioning. You have sugar the same way, you have proteins, you have everything functioning the same way. Even though the dark green here comes from a sick plant, they don't come from a healthy plant. And both are similarly different from sick leaves. When we do the same, looking at the transcriptome is all genes expressed. So we have methodologies to do that. And we analyze the difference in gene expression between these different situations. The same result, absolutely the same result, which means all sane leaves behave similarly. We cannot distinguish them. And all sick leaves, I mean symptomatic leaves, are different from the two other groups. And maybe I can illustrate that.
Bueno, ahí lo que estaba explicando es claramente que cuando hicieron el estudio, tanto a nivel metabólico como a nivel de expresión de genes, no pudieron diferenciar las plantas que no estaban afectadas por ESCA de las que tenían ESCA pero eran asintomáticas, es decir, que no presentaban el fenotipo de la enfermedad. Y sí se diferenciaban grandemente las que tenían el síntoma de las que no lo tenían, tanto a nivel metabólico como a nivel de expresión génica.
So this can be represented in different ways. So we call, when genes are not expressed in the same way in two situations, we call that differentially expressed genes. So I will just use DEGs. It's not nice, but this is the way we say it. So if you compare the control to the normal leaves of the sick plants, you know, we have no DEGs. I mean, two, that's nothing. Although the plants are different, and really they are 5 meters away, so these are very different plants. But when we do the same analysis between the sick leaves on the sick plant and the control of the sick leaf on the sick plant and the sane leaf on the sick plant, then you see we have roughly the same differences. So on the same plant, the difference between sick and non-sick is the same than the fact that you take the leaf from another plant. In other words, this leaf and this leaf, again, are very similar and both similarly different from the symptomatic leaves.
Claro, en este gráfico está expresando más o menos lo mismo, es solamente que lo está viendo a través de del DEG, que son los genes que se expresan en forma diferencial. Y se ve el mismo comportamiento, que la planta que estaba enferma pero no presentaba síntomas, no expresa genes, al igual que la control, y cuando compara la que sí presentaba síntomas, esa sí expresa y las otras dos se siguen comportando de la misma manera. Se comportan iguales.
So then we want to analyze the methylome. So I just make a short technical thing so that you can really understand what we have been doing. So what we study here is what we call the methylome, the methylation landscape over the whole genome. And we have a methodology to do that, which is called whole genome bisulfite sequencing. So we sequence the whole genome. So this is classical for genomic studies. But before we treat that with bisulfite, which has the specific property, it will transform a cytosine into thymine. In fact, into uracil, but after PCR into thymine, when the cytosine is not methylated. So which means if you have in a genome a mixture between methylated cytosine and not methylated cytosine, all those non-methylated C here will be transformed into a T after a while, but those methylated will be protected from that. So we will change the genome, and then we can, after sequencing, determine which were changed and which were not changed. So it's a bit complicated in plant because any C you find in a genome can be methylated. It's different from humans where only this CG methylation here can be methylated. We have all these situations. And then once we have done that and sequenced and analyzed, there's a long bioinformatic process behind, then we can analyze the distribution of methylation all over the genome. You will have things like that. You see cytosines methylated, not methylated. This represents a percentage of methylation. So this is nice to know, but not very useful as an information. So usually we try to determine regions that are differentially methylated. So not one single cytosine, but a bunch of them distributed at one place. And this is called differentially methylated regions, in other words, DMRs, and it's what I will speak now on. And this we can check that on the whole genome. And this will be some type of landmarks on all the genome that can associate with gene and so on and correlate with gene expression.
Bueno, el estudio que hicieron a nivel de la metilación, para saber, digamos, cuáles estaban metilados y cuáles no, lo analizan el genoma completo con esa sustancia bisulfito. ¿Qué particularidad tiene esa sustancia? Que las bases de citosina que no están metiladas las puede pasar a timina, pero las que sí están metiladas no las transforma a timina. Es decir, esa sustancia hace que todas las C no metiladas se transformen en T, pero las metiladas no se transforman. Entonces después lo que pueden analizar es la distribución de metilación. Y en definitiva, en la última parte, la metodología que usan es la identificación de las regiones que están diferencialmente metiladas. Ellos no les interesa ver si una citosina está metilada o no, sino que les interesa identificar en todo el ADN las regiones que están diferencialmente metiladas. Y en esos gráficos, digamos, que están abajo, ellos pueden ver de alguna manera la proporción de metilación del ADN.
Okay, so just to make it clearer, here you have, let's say, situation 1, here situation 2. If you look at this part here, you see there's no methylated cytosine here. You have methylated cytosines here. So this will be defined as a DMR.
As what, sorry?
Differentially methylated regions,
perfect.
DMR, because it showed that in situation 1 you don't have methylation, in situation 2 you have methylation.
Claro, así se define la sigla de lo que está arriba, que es las regiones que están diferencialmente metiladas. La que no tiene nada arriba no está metilada y la otra sí. Eso es lo que se denomina una región diferencial.
Okay, so if we go back to our case study, you remember that when we compare these two types of plants here, I mean the control to the non-symptomatic leaves, there was no difference in metabolite level and there was no difference in transcriptome level. If we look at the methylation level, we find many differences, sufficiently to work on that. But then so this is the first thing. So normal leaves from one sick plant to a non-sick plant are similar, are no more similar, they have differences. And then we do the same analysis comparing the symptomatic leaves, so ESAS, to the asymptomatic one, whether they're from control or from sick plants. And you see we have roughly the same profile. I don't say we have the same intensity, we have the same profile, which means you have a very big enhancement of difference in methylation in one sequence context. I said what on top. So this situation here, you see you have a hyper change in methylation here. And here they're the same. It's not as extreme because this is the same plant and these are two different plants. And this may explain this difference. But in both cases, the red bar is increasing quite a lot. So I just conclude, so which means here we see the reaction of the plant to the illness.
Lo que está mostrando acá es cuando ellos hacen ese análisis y entonces detectan las regiones DMR, que son las que estaban diferencialmente metiladas. En el primer caso, que no había diferencias metabólicas ni transcriptómicas, ahora sí encuentran diferencias en estas regiones con las plantas asintomáticas y las controles. Y después cuando comparan las enfermas con las asintomáticas y las enfermas con las controles, ven que en realidad, si bien es más alta la curva, la columna roja, pero el patrón de metilación es más o menos parecido. Las que están enfermas realmente presentan las que presentan el fenotipo realmente presentan, ¿no es cierto?, una reacción debido a la enfermedad.
Okay, then when you have this, you say, okay, it's highly complex. You have, sorry for that. So you have a lot of things. So what is common? What does this represent, in fact? So then you can compare the different DMRs, the different situations, and you see you have things that appear common to different. This is common to all. This is common to these two. And so I will not comment on everything, but just focus. So what do we see here, this 807? These are specific difference only found between these two types of leaves, control and symptomatics. What do we see here? This is more than 1,000, those that are different between the control leaves and the symptomatic leaves. And what do we see here? We see differences that are present both in this situation and in this situation. So it means these differences are always differences occurring between the leaf from the sick plant when we compare the leaf to a normal plant. So whatever the phenotype of the leaf of the sick plant, these differences are always found with a normal plant. So they represent a signature of the sick leaves irrespective to the leaf phenotype as compared to the normal plant.
Bueno, en este estudio, digamos, pasándolo a este nivel, lo que hicieron fue determinar 3 tipos de situaciones. La primera, que era diferencias específicas entre las plantas que estaban enfermas pero eran asintomáticas y las control. Esos eran, bueno, 807, no sé si son casos. En el segundo gráfico lo que ven es diferencias específicas entre las control y las que sí presentaban el fenotipo de la enfermedad. Y el tercero, que ese es, digamos, lo importante, eran las diferencias que se encontraban entre las que tenían la enfermedad con la control. Es decir, independientemente del fenotipo, las que tenían la enfermedad de ESCA, ya sea con fenotipo de enferma o con fenotipo sintomático, diferentes de la control. Y bueno, eso sería la señal de metilación de las plantas, independientemente de la expresión de los síntomas. Es decir, presentaban, ¿no es cierto?, una señal de metilación, independientemente de que el fenotipo fuera enfermo o sano. Y ese sería el marcador epigenético de las plantas que estaban enfermas con esa enfermedad. Es decir, que por más que presentaran o no presentaran síntomas, se había encontrado un marcador a nivel epigenético que te decía, esta planta está enferma.
So that was surprising because then we need to think what it means. So either it means that we have a systemic signal in the plant that modify the epigenome everywhere, or that, so it could be anything, or that you have toxins moving into the plant that could affect it. So whatever, it means that the activity at the epigenetic level of the illness is not local, it's affecting the whole plant.
Claro, eso es como lo más interesante, que por qué se encontraba un marcador epigenético en las plantas enfermas, o porque ese marcador epigenético era sistémico, es decir, se podía transmitir a toda la planta, o porque había toxinas o algo que se estaban distribuyendo en toda la planta. ¿Qué fue lo último que dijo? Que no lo retuve. What was the last part?
So either it's a systemic signal,
Yes.
or can be toxic,
Yeah.
a toxin which is migrating.
Yes, I said that.
No, but I said that.
Bueno, básicamente lo que dijo es que lo que sí es como importante destacar, o lo que fue choqueante de esto, es que la planta enferma tenía un marcador epigenético independientemente de los signos que se transmitía a toda la planta, claramente.
Okay, so we hypothesize from here that maybe we can find some very specific markers of SK infected plants. But of course, I mean, this is not we are in the situation, plants have symptoms, so it's too late. There's no memory there, it's simply what happens when the plant is sick. So then we designed do you want to translate it? You need to translate now.
¿Eso? No, bueno, ellos lo que querían ver es que si había realmente un marcador para la enfermedad, pero era tarde con una que ya tenía los síntomas, es decir, ya era obvio que iba a tener algún marcador.
So there's no memory here.
Claro, no había memoria.
We are during the response. So then we designed a different experiment. So I passed that because we just said it. So we designed a totally different experiment with the same question. So we went back to the field and checked for plants that have expressed during the previous years. I mean, since here, 2018, they have expressed symptoms 4 times, 5 times, 4 times, not always that often. And some other plants, similar situation, very close by, that have never expressed symptoms during the 4 years. And we said that if what we found before is related to any type of memory to the SCAT, then we should find in this situation the same type of marks.
Bueno, pues entonces dijeron, bueno, vamos a plantear otro trabajo en donde tomaron 2 tipos de plantas, que eran plantas que habían sido trasplantadas en maceta y donde sabían su historia. En un caso, las que están en rojo eran plantas que habían presentado síntomas 4 veces, 5 veces, y en el otro caso, las en verde, plantas que nunca habían presentado síntomas.
So we took this plant, replanted them in pots because we wanted to do some other experiment on that, and decided, and we did this 2 years in a row. First year, just to control, but you have the stress of reporting, so we don't know what exactly could come out, but still we use them, and then the second year. The difference here is that we used only very young leaves. We didn't use leaves that were already expressing symptoms. So whatever analysis we did, then they were on leaves much before any symptom could be expressed.
In both cases.
In both cases, for all these plants, it was early during the growing season, so the plants are perfectly well. You don't see any symptoms. Maybe they will develop later, but for that we don't know. And we did the exact same experiment.
Lo que hicieron para reproducir el experimento fue de esas plantas que históricamente habían presentado síntomas y no, eligieron esas 3 hojas y las reprodujeron, pero no sacaron hojas que estaban enfermas. De los 2 casos, sacaron hojas jóvenes que no presentaban los síntomas, independientemente de que una venía de plantas que había presentado síntomas y la otra que no. Lo hicieron en maceta.
And so the idea is that can we find epigenetic marks related to the symptoms without symptoms on these plants? So this would be some type of memory markers.
Claro, y la pregunta era, porque en la otra ya tenían los síntomas, en cambio en esta lo que querían ver, ¿podemos encontrar estos marcadores de memoria de la enfermedad por más que no las plantas que pusimos las tomamos de órganos que no tenían síntomas?
So I will not go too much into detail. So this is the previous result we had, these 554 that were common and reflecting the difference between the sick and the normal plant. And so I don't redetail the experiment, but when comparing 2018 and 2019, for this plant, we found, 832 and 17, 715 DMRs, and then we cross all data. So the idea is, are there some here common to those and to those? Knowing that we have been doing a lot of different things. So this is the outcome. We found 5. So it doesn't mean a lot. That's not a lot. But if we look at the 5, so 5 is not a lot, but it's a lot at the same time, because often you will not find any. So which mean, and the point is that when you look at the 5 DMRs, and okay, some are unknown gene, but 2 of these genes are really involved in the response of the plant to the pathogen. So one is the F-box family, which is a gene family which is induced in response to biotic and abiotic stresses. And the other one is the ascorbate oxidase. You all know that ascorbate regulation is involved in the redox homeostasis during stress response.
Bueno, entonces el resultado que obtuvieron es que de los 554 y los 832 zonas de metilación diferenciada, bueno, solamente encontraron 5, que dice que puede parecer poco, pero que hay veces no encuentran nada, y que analizando esos 5 genes encontraron 2 que están o estarían íntimamente relacionados con esta enfermedad. Uno es el de la familia de las proteínas F-box, que tienen que ver con respuestas de la planta a factores adversos, y el otro es un gen relacionado con la senescencia que tiene que ver con la ascorbato oxidasa. Esos serían los 2 genes de importancia que identificaron que podrían estar relacionados con la enfermedad.
So at the end of all these studies, we find out that 5 possible methylation marks are associated with the illness. Plants that have developed or will develop the symptoms do present one allele of these DMRs, and plants that do not develop will present a different one.
Claro, entonces lo que encuentran es que las plantas que sí presentaban, ¿no es cierto?, los síntomas de la enfermedad, presentaban al menos 2 alelos con esos genes, y las que no presentaban no los presentaban.
So this correlation leads us to the idea that they are indeed marks, and then I will conclude on that. So finally, what we have been showing here is that although we don't find differences in metabolome, transcriptome for same leaves, whatever the origin, we can find that the symptom provoke a remodeling of the both type of leaves, actually, whether they are normal or not. So we have a systemic effect. We also show that, and this is the second experiment, the SK modified epigenetic landscape is maintained over years through the different generations. And then we have found 5 possible markers that are associated with the illness in one form and not with the illness in another form. With one more question now, is this really a memory of the stress, or is this a marker of the susceptibility to the stress? So we are working on that. So what we do now, we develop different approaches to look at wide population and check systematically for the presence of these different forms of alleles and correlate with the illness.
Bueno, ¿cuáles son algunas de las conclusiones del trabajo? Que claramente la infección de la enfermedad ESCA lleva a diferencias, modificaciones metabólicas y transcriptómicas solamente en plantas o en los sectores de planta que expresan los síntomas. Es decir, que a nivel metabólico y transcriptómico no habría diferencias entre los controles y las que eran asintomáticas. Después que la infección con ESCA lleva a la remodelación del, digamos, a remodeling methylome. What's remodeling methylome? Ah, bien. Claro, que en realidad había, ese era el gráfico que mostró que sí había diferencias en la metilación en todas las partes de la planta que estaban enfermas, independientemente de que si presentaban o no el fenotipo. Ahí es donde habíamos hablado de que había un efecto sistémico. Después lo que también vio es que algunos cambios epigenéticos que se producían con el ESCA se mantenían en las plantas a través de los años y que estaban asociados con la expresión, ¿no es cierto?, de los síntomas de ESCA y con la historia de la planta. Entonces la pregunta es, ¿son buenos marcadores estos para determinar la infección con ESCA o en realidad son marcadores que están expresando la susceptibilidad que tiene la planta? Bueno.
Okay. So this is, I mean, just to conclude on this, so you see that through epigenetic information, we could define something that we could not define using either classical phenotyping. Phenotyping of a leaf is very late. Here we can phenotype the plant very early, I mean, before any symptom, you need to wait months. So we have access to information that no other approaches could provide. So that's really a strong added value.
Can I ask a question about that? Does mean that you can like analyzing the epigenome of those plants or those markers can predict if a plant is going to be okay. Lo que le estoy preguntando es si en realidad con ese approach que él está diciendo, bueno, se podría predecir si una planta se va a enfermar o no con ESCA o está enferma de ESCA, por más que no presente síntomas, analizando esos marcadores.
And then we have harvest going in different lots, 1 yard where it's quite difficult. I'm sure why you see the correlation. Okay, are you still alive? So just to make briefly another part of this epigenetic practice, another experiment done by my PhD student. We have been trained to modify the carbohydrate balance. So this is not a field experiment.
Sorry, this is not a field experiment. This is an experiment designed to stress the plant. It's not to maintain the plant. So just don't be shocked of what we have been doing to the plants. The question was to study the transannual memory, but then you see it will be evolved in different ways. So we worked on a population of plants that have been grown in pots for 8 years. They were all the same. And the idea was to control the carbohydrate level, but a dramatic canopy reduction of the plant. So we really killed them almost. And then we did that several years, I mean, 4 years exactly.
Bueno, en este estudio lo que hicieron fue no tratar de que las plantas estuvieran bien, sino a propósito les provocó un estrés muy grande, un estudio en plantas que estaban en maceta por 8 años, creo que dijo, y lo que hicieron fue hacer una gran reducción de parte de la canopia, me imagino que serán hojas principalmente, para analizar bueno, la memoria que tenían, ¿no es cierto?, a esa limitación de carbohidratos, en definitiva, porque le estaban sacando las hojas. Pero la memoria que tenían transanualmente, es decir, al otro año o en los años venideros.
Okay, this was the last part of the memory. I mean, those of perennial plants, what happened when you analyze year after years, how the plant evolves. And we have the same type of approach, which means we integrate data from epigenomes, genome, transcriptome, metabolome, phenotype. So we integrate all type of data.
Hicieron los mismos tipos de análisis que en el caso anterior y después los integraron.
Yeah, and we try not to focus. This is a blind approach. So we check everything as much as we can.
Y trataron de no focalizarse en nada específico, sino en un ensayo a ciegas, digamos, en general, en todas las plantas.
So this is the experiment. So we started in 2018. So we had 32 plants, all equivalent normally. So we randomized them because there was no differences. And then on some plants, we let 12 leaves per bunch, which is quite a lot. I mean, I thought. And on some others, we let only 3. That was really not a lot. And then we let the plant grow. So we control the canopy the first year. Before doing that, we have harvested leaves for the analysis of the plant before any stress, though the initial state to compare. And then when we have done that, the year after, so again, before any stress, we have harvested leaves. And then we have looked at the phenotype the year after. So the control plant, they look pretty nice. And the surprise came from those plants here, those with only 3 leaves. Half of them, it's random, it was really half of them, had almost normal behavior. They were less robust, but they could form fruits normally, they could develop normally, and half of them were like this. So which means no fruits, very little development, so they were suffering a lot. Although, and this is the point I want to stress, when we look at the plant here, there was absolutely no phenotypical differences. So we were unable to predict in any way this behavior.
Bueno, el estudio en realidad era bastante simple, eran todas plantas que no tenían ninguna diferencia fenotípica, todas iguales. En un caso les habían dejado 12 hojas y en el otro caso les dejaron solamente 3 hojas. Pero previamente a cortarles las hojas, recogieron hojas que provenían de las plantas antes del factor de estrés, digamos, antes de someterlas a ese factor de estrés. Al año siguiente, esas plantas que produjeron, bueno, las control, digamos, produjeron plantas normales, que sin ningún problema, y en el otro caso, 2 tipos de plantas. Unas que eran relativamente normales, un poco más pequeñas que las otras, pero relativamente normales y con fruta, y otras que no, que eran directamente muy limitadas, sin fruta y con muy pocas hojas.
So and again, here you cannot separate them, they're all the same.
Arriba
Really.
no las puedes separar, son todas iguales.
So this behavior is unpredictable. It means that finally you have into an homogeneous population of plant, and I think you see all that in fields. Population is homogeneous, something happened, and all plants will react differently. So this is classical, but at least here we can see it very nicely.
Era impredecible el resultado, porque habían partido una población totalmente homogénea y bueno, no se podía predecir cuál iba a ser el resultado después.
So we did that 3 and 4th years now. So this is typically those that were affected dramatically, though they were less affected and control. But over years, what we have noticed is that the phenotype of the fruits were changing. So the first year, those forming fruits, so not this one, but those one or the C3 with fruits, progressively the ripening was altered and modified. And the last year, the C3 plants with fruits did not ripen. So there was a problem, obviously, of sugar providing that will enhance year after year.
I have a question, maybe I didn't understand. When you reproduce the essay, every year you cut the leaves or you just cut the leaves once?
Every year.
Ah, ok. Bueno, lo que está mostrando es que a medida que lo iban repitiendo, encontraron diferencias en el grado de maduración de la uva. Las plantas del control siempre maduraban normalmente y las otras, a medida que avanzaban los años, iban madurando cada vez menos, de peor manera, hasta que, bueno, no sé si en el tercer año directamente no lograron llegar a maduración.
So I think this is quite classical about the fact that when you have less and less sugar. So, but it shows a cumulative effect over years, which can be a different origin. You don't store enough sugar, or you have some additional factors.
Claro que ahí es un efecto como que se va sumando año a año y que puede ser simplemente porque no hay suficiente azúcar para llegar a esa maduración, porque hay menos hojas, o porque hay otro efecto que se va acumulando con los años.
Yeah, this is Cabernet Sauvignon. Okay, this is just to summarize. So first year we harvest here, and then we do the canopy control. Then we split. I mean, this one is the control and no effect. Then we split those plants in two groups: those not developing a lot and no fruits, and those developing fruits and almost normal. When we of course, this one we cannot do anything more. I mean, if we do anything, they die. So we let them recover. But they understood this one very rapidly, that they could rest in peace. But this one that was still forming fruit will reproduce the same treatment. And then we see that some will join the group here. And those that have recovered will join the group here, but not at the same speed, not all. So they are very different from each other. So each plant is very specific. There's no general rule for that. And then we go on and still we have the same kind of scenario the year after.
Bueno, entonces ahí está demostrado lo que hicieron los 3 años. Las controles siempre se mantuvieron iguales y lo que sucedió con las otras, las habían separado en las que producían fruta y estaban más o menos normales y las que no. Como estaban tan mal las que no, las dejaron recuperarse, no les siguieron cortando las hojas y lo que vieron era que las que venían estando bien de repente pasaban al grupo de las que estaban peor, es decir, dejaban de tener fruta y tenían menos hojas, y las que se habían empezado a recuperar, las que le habían dejado de poner el tratamiento, también saltaban al grupo de que, bueno, mejoraban, volvían a tener fruta y presentaban más crecimiento.
Okay, so now just to explain what type of analysis. So first, you have to have that very clearly in mind. We never analyze after the stress. We just analyze the leaves before the stress is applied. So each time we look at the effects of what has happened the year before, not what is happening during the year. So this is before the first stress, this is before the second stress, and this is before the third stress. So we are only interested in the memory of what has happened the year before. We are not looking at the effect of the stress.
Claro, para poder realmente ver los resultados que ellos querían, no analizaban las hojas después de impuesto el estrés. Siempre, cada año, las analizaban antes de imponer el estrés. ¿Por qué? Porque ellos querían realmente averiguar el efecto de la memoria que habían tenido de lo que les había pasado el año anterior.
So this is another way to represent gene expression analysis. So this is the same thing than before. Just to help you to read that, each bar here is a gene, and the color indicate the level of expression of the gene as compared to.
Sorry, the color?
Indicate the level of expression, highly expressed, red, lowly expressed. So this analysis before any stress. Here you have the plant that were treated after C12, C3 that did not produce fruit, and C3 that still produce fruit. The question was, are they organized based on the phenotype the year after? Is that clear? The question, so when we compare the gene expression between these different plants, do we find all green together, all red together, all blue together? Because these plants will have different behaviors. And so when we do this analysis before any stress, you can see they cannot be separated. They're all mixed.
Bien.
Okay.
Claro, que el primer año que les habían sacado las hojas antes de cualquier tipo de estrés, no podían diferenciar a los 3 tipos de plantas, si estaban todas mezcladas.
No, I have 5 more.
Y después sí las podían diferenciar en cuanto a su expresión génica. Antes no las podían diferenciar.
It's not possible here.
No era posible.
But then the year after, so again, here we analyze the plant before the stress of the second year. So we see the consequences of the stress that occurred here. And now when we look at this, you see there were 2 different panels. All those are the plants that were not able to make fruits here. And all those were either the control or the plants that were able to make fruit after the stress. So we now very clearly separate them simply based on the phenotype before they express the phenotype.
Claro, y ya en el segundo año que les cortaron las hojas antes de, analizaron las hojas antes de ponerle el segundo estrés, pero ya habían tenido un estrés el año anterior, se podían diferenciar perfectamente. Las que no producían frutas y eran chiquititas de las otras 2, las control y las que les habían sacado hojas, pero iban a presentar el fenotipo, digamos, de casi normal. Es decir, que ya tenían una diferenciación antes de someterlas al nuevo estrés.
Okay, I don't comment the next year, it's more complicated. Okay, then we did the same thing for the epigenetic analysis. So this, I don't go into all details, but same leaves, same samples. We extract RNA and DNA, and then we separate the analysis. If you remember well, I show you again. So here we couldn't distinguish the plant. They were all equivalent. Now we look at the methylome. So here we compare this one to this one. But before here, you see they have differences. If we do this one with now, this one, but the year before, they have similar level of differences. But if we do this compared to those the year before, okay, then you see they have much more differences. So which means the plants before they were stressed, the 2 group of plants that we have called C3F0s, which mean no fruit after stress, or C3F fruits after stress, these 2 group of plants are very different as compared to all other comparisons we have been doing. So which means the epigenetic landscape here is sufficiently different and reflect the difference in behavior after.
Claro, bueno.
Was clear? Oh.
Sí. Lo que vieron después al analizar las regiones estas de metilación diferenciales, que es lo que les marca el cambio epigenético, era que las plantas que antes de someterse nuevamente al estrés, pero que ya habían tenido un estrés el año pasado, eran muy distintas a nivel epigenético las que no iban a presentar fruta de las que sí iban a presentar fruta. Presentaban una diferencia en esas regiones de metilación que era muy grande.
Okay, and so this reflects the fact that the plant will behave very differently after.
Eso a nivel epigenético refleja el hecho de que después el comportamiento de esa planta, que es lo que después ellos vieron, iba a ser muy distinto.
Okay, and very briefly on that, then when these plants then are treated differently, the difference will increase with time. Okay.
Y esas diferencias, a medida que eso seguía sucediendo, iban a incrementarse con el tiempo.
Okay, just to summarize this. So first thing, and for us non-viticulturists but plant biologists, it was surprising that 2 plants treated in the same way have very different behaviors. So it's what we have shown here, the first thing. We cannot separate plants before any stress based on, I didn't show the metabolic data, it is the same. We cannot separate them on phenotypes, metabolites, transcriptome. They are all similar, but we can separate them based on epigenomes. So which means the internal information provided through their life and that progressively created a specific epigenome allows us to differentiate and to correlate that with a specific phenotype after. Okay, these are different conclusions. I didn't comment on that.
Bueno, entonces, ¿cuáles son las conclusiones? Que claro, que en realidad el control de la canopia llevó a respuestas contrastantes, pero en realidad esas respuestas dependían de un estado inicial de las plantas. ¿Por qué? Porque ellos la primera toma de muestra para analizarlo lo hicieron antes de poner el estrés. Es decir que stress allows selecting the plants, es decir que al someter al estrés de esas plantas, bueno, esas plantas respondieron de acuerdo a un estado inicial que tenían, evidentemente a nivel epigenético, porque no lo mostró, pero cuando ellos se basaron en el análisis metabólico, fenotípico, transcriptómico que tenían esas plantas antes del estrés, eran todas iguales, no había manera de diferenciarlas. Entonces, el escenario de metilación que tenían permitió discriminar esas plantas en base a qué? A una respuesta distinta de ellas al estrés que se aplicó antes de aplicárselo. Eso es lo interesante. Es decir, que ya ellas tenían estas diferencias de metilación y que cuando se aplicó el estrés dieron por resultado diferencias fenotípicas.
So we'll just conclude now. So this is what I mean, the objectives global. So and this is a question we are addressing through this experiment. So what part of the epigenetic variation generated by stresses are maintained? So what is the transannual memory? Then in addition to that, so what epigenetic drift will be created, and then how this epigenetic drift can be related to evolution of phenotypes. Second question is, can this epigenetic drift be genotype dependent, which means different cultivars will not behave in the same way over years? And finally, can we link all this to one typicity? So is there a link between the evolution, progressive evolution, and this is the question related to terroir, in fact. Is there a link between the evolution, epigenetic evolution of plants and the fact that different plants in different environments will have different epigenetic evolution with time, sometimes leading to death? This is the grape decay we have in Europe, which is dramatic, sometimes not. And can this have a link, direct link with one typicity? I skip this one.
Entonces, bueno, ¿cuáles son las preguntas, no? Bueno, ¿qué partes de las variaciones de la metilación que se inducen por un estrés se mantienen a través de los años después que ese estrés se eliminó, digamos, se sacó? Es decir, ¿cuáles son esas que se mantienen? Podemos evaluar Luciano, ¿cómo traducís drift?
Drifts, evolution. Drift, c'est la dérive. The fact that it's derived, I mean, it's.
La deriva.
Yeah. So,
Ah, claro. Es decir, podemos evaluar la deriva epigenética, es decir, cuáles de los cambios epigenéticos se van a generar por diferencias en el medio en distintos genotipos. Genotipos, estamos hablando de individuos. Podemos determinar la relación que hay entre el fenotipo, el genotipo y los efectos epigenéticos que han sido producidos por el ambiente, y cuál es la relación entre el estado epigenético inicial, la respuesta que va a tener ese individuo al estrés y, en definitiva, el fenotipo que va a presentar. Estamos hablando de una planta de vid, ¿no? Entonces eso tiene un impacto muy grande en el fenotipo y la tipicidad que puede tener ese vino o esa uva en un terroir específico, que el terroir específico, entiendo, es el que le va a dar los cambios en el medio ambiente.
Okay, just to conclude, so these are the people who working in my group. So those that have participated to the work is Margot Berger. She has been doing a PhD on the canopy control. Linda, she's the main operating person. I mean, she's an engineer doing most of the thing in the lab. She's very precious. Bernadette, she's specialized in bioinformatics, and she's been doing most of the bioinformatics. With Virginie, Emeline is one of my long-lasting collaborator, and myself, and those people are doing other things.
Okay. Thank you very much, Philippe. ¿Alguna pregunta?
Buen día. Tengo entendido que las plantas, por lo que veníamos hablando, las enfermedades epigenéticas llegan a descubrirse por lo general cuando son asintomáticas, cuando dan los frutos, o puede ser por medio de las hojas. Yo digo, estas enfermedades por epigenética, cuando se van recuperando las plantas, ¿se pueden recuperar al 100% para llegar a dar una misma calidad de vino que las otras que no tienen esa enfermedad por epigenética? Y si alguna vez también estas plantas pueden llegar a haber algún caso extremo en que se las tenga que sacar. Nunca escuché que hayan, por ejemplo, después de 5 años cortado una planta, o sea, sacarla y poner directamente otra. Esa es mi duda, no sé si es... que si por alguna enfermedad epigenética no se lleguen a recuperar al 100% las plantas. Esta es mi consulta, si se puede llegar a dar que no se recuperen, que no den un vino de la calidad deseada y que se pueda llegar a sacarla. O es que sí se pueden curar estas plantas y llegan a la calidad deseada.
La respuesta
Well, I know it's quite complicated. The thing is, and I think that you can relate it with the tomatoes that then turn red again. The epigenetic changes can be like reverted. For example, in the case where you explain the plants infected with ESCA and some leaves were symptomatic and some were asymptomatic. A symptomatic leaf in the future, a plant from the symptomatic leaf can be asymptomatic again if you Bueno, la primera parte es que los cambios epigenéticos pueden ser reversibles, sí. A diferencia de los cambios a nivel del genoma, los cambios epigenéticos sí pueden ser reversibles. Claro, y con respecto a lo que vos preguntaste, en realidad, si la planta sobrevive o no sobrevive, o si los síntomas pueden ser menores o reversibles, eso no depende de la epigenética, eso depende del tipo de enfermedad de la planta y de su consecuencia en la planta total. Lo que sí ellos están evaluando es, bueno, el grado de reversibilidad del cambio epigenético o no en la planta. ¿Alguna otra?
Sorry for my English. I have two plants
Pinot Gris that express two colors in the clusters. Could it be the virus?
Ah, that's very interesting. Okay, so that's nice. So you know what the color change is due to. This is usually due to a mobility of a transposon that will move into one of gene that control the accumulation of anthocyanins. I mean, I don't remember. I think it's MIB-A1, which is the gene targeted. So I didn't speak of that. I had no time, but if now that's quite nice actually. So transposons, which are, I mean, you know what a transposon is or you don't. So these are a small piece of DNA that can jump. They can be mobile into the.
Primero la pregunta de él fue que él tiene que plantar dos Pinot Noir que tienen, Pinot Gris, que tienen distinto color, expresan distinto color en el racimo. Ah, en una misma planta.
It is the same plant.
Ah,
Yeah,
perdón.
that's
Sí.
exactly.
No, pero la gente no lo había escuchado la
Okay.
pregunta. Decíla en castellano.
En Valle Templado tenemos una plantación de Pinot Gris que de 100 plantas hay 2 que en la misma planta tienen racimos de 2 colores. Perdón, no racimos de 2 colores, sino el mismo racimo está partido en 2. Y la planta tiene un virus, no sé qué virus será, pero bueno, la pregunta es si se debe realmente a ese virus. Y bueno, este año las hemos reproducido, vamos a ver cómo se expresa.
Él te estaba diciendo que el transposón es una parte del ADN que tiene la capacidad de saltar de un lugar a otro del genoma.
Yeah. So you have a question. It's always, I mean, so on one plant you will have different bunch, and they all have this behavior, or it's different. You have different types of phenotypes.
¿Todos tienen eso o que en la misma planta algunos racimos y otros no?
No todos los racimos. Algunos racimos y otros.
In the same plant, not all the bunches, but some of them.
Some of them, and when they present, it's always the same.
Ah, no, es la primera vez que lo presentan.
Okay, so this reminds me really of the behavior of transposons because these are jumping randomly at different times, and when they jump in the gene, they kill the gene. So typically, this is what happened for the difference in color. I mean, for grape wine, in white and red are due to the insertion of a transposon into the C1 gene. And this causes the change in color because you impair the ability of the plant to induce the gene. So you have this. It seems to be unstable in your case because otherwise all will be the same. So it means that sometime during the bunch of development of your bunch, you have transposon jumping. Need to do more study to determine which one and why. But typically that is jumping, and then when it jumps into the gene C1, I think I know the transposon because there's one doing that. When it's jumping, you kill the color.
Lo que dice es que una vez que se produce este salto del transposoma, como que mata la capacidad, mata el gen que tiene la capacidad de acumular el color. But the question is now he is multiplying that plants somatically. So because he would like to know if
I guess he will have the same thing. So because the question is, it's not that the transposon is jumping, it's why it's jumping. And so there's a loose control on this transposon. It means that it should be controlled and it's not. And normally when it jumps, then you have a stable white color, and it's not jumping, then you have a red one. When it's moving, the control of the system is loose. So sometimes it's moving, sometimes it's not. And So that's not stable. So which means the regulatory processes before have been affected in some way. So maybe you have a variant. You can send me some plans.
Claro, dice que en realidad él piensa que se te va a volver a producir porque no es el tema de que salte, sino de el modo en que salta o por qué salta. Es decir, que se ha perdido la regulación de ese proceso y eso se va a transmitir.
And do you have grains that are different colors? Only part of the bunch. So it's very early.
Just the cluster.
Yeah. So it's a very early event. So it's even earlier. It's not during fruit development. It's probably during flower meristem development and things like that. Yeah. 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 But it is a mixed bunch, or you have one bunch and another one here. The red behind, how is it? Is it the same?
Si son racimos mezclados o si es el mismo racimo.
No, el mismo racimo.
The same. So yes, it's really during the flower, I mean the meristem development of this bunch. Yeah. Yeah, it's quite interesting.
¿Alguien tiene alguna otra pregunta?
Bueno, lo que le voy a preguntar a Philippe es si él hizo toda su investigación en Cabernet Sauvignon y si cree que hay un efecto varietal en la memoria de las plantas. Well, you have done your entire research in Cabernet Sauvignon, I don't know. And
not always. I mean,
okay.
we have some work on Cabernet Sauvignon, some on Semillon, some on Pinot Noir, some on different types of genotype have been analyzed.
And do you think that there might be a variety effect in the memory of plants? Have you seen a difference between the varieties?
So I will not say it this way. I think the alleles of the gene that control, I mean, the epigenetic processes have an impact on what happens, and this can affect the memory. So it's really all the machinery behind that determine that. It has been well shown in other plants in Arabidopsis. Different alleles of specific genes cause different phenotypes, and I mean, on the long term, adaptive capabilities. So I guess in grape one, it would be the same, but it depends. And there's no knowledge about that so far in grape wine.
Okay, so there might be a difference.
Yeah, I guess yes.
Okay.
I mean, I'm not saying there's a difference in the memory. I think there's a difference in the reaction of the plant. Okay, because the allele, I mean, the mechanism controlling all these things is different because you have allelic variations. But then when we come to the memory itself, which this is very basic replication processes and so on, I think it's going to work pretty similarly, but what is set up first might be different. Okay.
Bueno, sí puede haber una diferencia varietal, pero que no está expresada. Yo lo término genético me lo salteo. No sé si lo crees. Claro, el funcionamiento de las plantas es igual, pero... ¿Alguna pregunta? That's all. Thank you very much, Philip.